want to begin by having our two panelists introduce themselves, and then we'll go back to me and I'll give you your first topic. Okay, Irina, why don't you go first?
Hello, everybody. Looks like great summit already. So I'm Irina Conboy, and I used to be professor of bioengineering at UC Berkeley for 21 years, until a month ago or so, when Alina Su, who is Chief Executive Officer of Generation Lab, finally convinced me to switch gears and to join as Chief Science Officer of Generation Lab. Before that, I did my PhD at Stanford in molecular and cellular immunology, and after PhD, I did postdoctoral training with Tom Rando, who is now at UCLA, wonderful scientist, and I wish he was here.
Denis?
Okay, good morning, everyone. My name is Denis Evseenko. I'm a professor and vice chair of orthopedic surgery at USC Keck School of Medicine. And I'm also a founding director of New Longevity Accelerator at USC, funded by a $10 million gift that we received a few months ago. And the goal of this accelerator is to help academics to advance their innovations into clinical space. So we operate primarily at the USC ecosystem right now, but looking forward to expanding that. And also I want to mention that I have two startup companies, both at clinical stage, focused on osteoarthritis and joint health. But we still face a lot of regulatory challenges that I'm looking forward to discussing today with all of you. Thank you.
That's a great transition into the first thing that I'd like to talk about, and it's sort of a stage-setting question, and maybe to spare each of the speakers in the talk sessions a slide or two about context, which is, for those of you who are new to longevity biotech, one of the things that we talk about a lot in our field is this critical challenge, was that the FDA doesn't recognize aging itself as an indication, that is to say, for a thing for which you can develop a drug to treat and run a trial for. So instead, you have to focus on a specific disease. So from the standpoint of founders, from the standpoint of people who are trying to help academics transition or translate their findings into biotech, like, how should you approach the FDA indication dilemma to actually get therapies to patients? Denis, why don't you kick us off?
Yeah, I think there's multiple layers to this problem, right? So the way how the FDA functions, I just came from Boston a day ago. That's why I don't have my voice, because it's so cold in Boston and raining. But I met with few people from the FDA, and the same thing again and again, right? So they really want to go after specific indications, right? In our case, it's osteoarthritis, which is a major indication. And it's age-related, right? So in vast majority of patients, this disease happens after the age of 65, affecting 600 million people worldwide. The problem is that there's no a single drug approved yet for osteoarthritis, right? And one of the reasons is because people are treating only joints, right? So they're not really treating the patient. And we talk a lot about multimorbidity these days, right? So if you think about a 65 or 70-year-old patient with advanced osteoarthritis, this patient will probably have other 3, 4, 5 diseases, right? Some of which will be systemic diseases. Probably obesity, diabetes type 2 involved, cardiovascular issues, etc. So if you come to the FDA and say, can we run a trial and look at some of this other, diseases in this patient, they will not let you. So they will force you to just do the joints and look at things like total joint space narrowing, pain. They're not going to let you look at other things. And I'll just give you one example. About 2 studies published this year. One of them is a retrospective study done by Novartis, where they report that patients treated for example, cardiovascular diseases, and that's a striking thing, with interleukin-1 receptor antagonist. So the Novartis actually did this trial, and they didn't meet their primary outcomes. So the drug was basically discontinued, and the study was considered unsuccessful, right? But after that, retrospectively, they analyzed this cohort of patients, and they found in a small group of these patients, specifically with systemic inflammation, had significantly reduced number of total joint replacements. but that was not looked at because that's how the FDA does their trials. When you only look at the joints, you miss what happened in the brain, in the heart. You only look at the joints, right? And then I gave you an example of a study when they only looked at the cardiovascular function and they didn't look at the joints, but it actually has worked for the joints. So I think what we need to do is to change the way how, I don't think we can change the way how FDA approves, looks at aging, and they're probably not going to accept it in the next decade or two as an indication, but we probably can change and convince them that aging is a multimorbidity issue and they need to allow companies to look really beyond one single indication. So that's how, in general, I looked at the problem.
I see Irina nodding her head. How are you thinking about this at Generation Lab?
Yeah, so first of all, it's a very good point that most diseases are systemic and we try to treat them as disease of one tissue. So that would be probably the low-hanging fruit to change FDA opinion on what do we consider success of clinical trials in a disease. But I also think that, and that brings about Generation Lab, is that we are rapidly exhausting our medical capacity to treat diseases. We actually made great progress in medicine. And that progress allowed us to ameliorate a number of diseases, but then there is kind of a plateau. Because when things are already broken in our body, it's very difficult to fix them. So another way to perhaps advocate to FDA, if instead of doing sick care, start doing health care. Before things are horribly wrong, there are also biomarkers of things are beginning to deteriorate. And there's new set of biomarkers, in fact, is what Generation Lab is expert in, allow them to inform doctors on what and how to do before a person has a critical illness. So approaching aging, not as just the process of natural decline, but as a process when various organ systems starts to be broken. They are not yet broken, you don't have cancer yet or diabetes. But things now are going a little bit less positively. So detection of that as the indication of aging progression and regression, I think is a great, great next point.
That's fantastic. And we're going to talk about biomarkers a little bit more in a second, but I want to stay with this idea of a healthcare system that's focused on sick care rather than healthcare. That is to say, you have to be a patient before you can get a medicine. And that has to do with a lot of things, but I think it has to do in part with some of the regulatory incentives or disincentives to try to get preventive medicines approved. And so I want to kind of open the floor. I know Denis has something to say about this. Why do you think it's hard right now to advance preventive therapeutics? And what might we do to solve that? You don't have to boil the ocean. You don't have to solve the world's problems, but I'm interested in your ideas.
Well, I think FDA is an evidence-based agency, and they're reasonable people, right? But it's very conservative and old agency. So I think we just need to build a stronger case. I think they're open to look into the concept of preventive care, and many of you will know that Metformin Group has convinced them that, you can do trials that, are designed to treat one disease and then look at the prevention of other diseases, right? So I think this concept is out there, and the FDA was open to it. So we are approaching it from the concept that we call arthritis plus concept, which is basically based on the premise that every single person after 65 will have osteoarthritis in one way or another, and so can be treated for this disease using a traditional path that FDA is very familiar with. All we need to convince them to do is to incorporate some additional outcome measures that will go outside of the osteoarthritis field and make them non-research outcome measures, but actually part of the study, part of the regulatory trial, because if we hit these outcome measures, so then we can make some claims with respect to our therapeutics. And that's how we are approaching it. So it's an arthritis plus concept. And we're also doing a big study on aging dogs. I'm happy to talk about it. Because I think, companion animals, and specifically dogs and cats, can actually help us to deliver this message across to the FDA. Because at the moment, there's no really good animal models of human aging. And we can talk about it if you want, but I'll stop here for now.
No, that's great, and I think that it's a great point that there are useful models, there are convenient models of aging, but none of them are human beings, you know, and mice aren't us, and we aren't mice, and it means that a lot of promising drugs that look great in rodent models don't look great in humans, and you know, dogs share our living environment, they share more of our diet than they ought to, right? And they've co-evolved with us for 10,000 years, you know, and so I think that they're a great model in which to study aging, and I think we could have an entire panel about that. But I want to give Irina maybe a chance to jump in on the prevention issue.
Yeah. So thank you, Chris. So I think that for prevention issue, we need to convince FDA that it is not something nebulous. We are treating healthy people, and this is all we do. So I like metaphors, as you will probably guess even later on from my talk. So we are flying on this airplane, and we know it's going to crash. It's 100% knowledge that the airplane is going to crash. And we have a kit to build a parachute. Why are we not using the kit to build a parachute? So that is my metaphoric understanding of applying completely novel concept of understanding aging as the fundamental cause of diseases, and also using prevention, because there is no true prevention, right? We are progressing towards being less healthy. So it is stop this progression towards the cliff while we can. So yes, I agree with Chris, I agree with Denis that FDA is very old, kind of, you know, and bureaucratic and set in its ways agency. But ultimately, they're our friends, and they want to help progress of biomedicine. So I think all that would be required is have meetings with them, workshops and meetings, and explain that we now can do much more if the regulatory landscape changed.
That's great. So one of the regulatory landscape changes that a lot of people in longevity biotech wish we could see is the use of aging biomarkers as legitimate endpoints in clinical studies. And so I think we need to do a couple of things in order to bring this concept to this group. One is we need to talk about what a biomarker is, like what do we mean when we talk about a biomarker in the context of a way to measure aging, and then maybe to frame that up a little bit more tightly, what would constitute like a credible biomarker development program that could actually support clinical translation? So again, let's define biomarkers and let's talk about their potential role in clinical translation. In Irina, given what you're doing for a living these days, I think you should start us off.
So yes, I'm very passionate about this because biomarkers of aging have been searched for many, many decades, even before I was professor at UC Berkeley, before like 2000s. And many excellent groups and scientists looked for them using hundreds of millions of dollars, and we did not find any. And now, to my dismay, I hear again exactly the same refrain. We need to know the enemy, we need to find the biomarkers of aging. So I think instead we need to ask the question, why are those so difficult to find? Do they actually exist? And to come to this point, what do we expect a biomarker to do? So for example, if somebody has propensity for diabetes, we look at the blood glucose levels, sugar levels. It's an excellent biomarker. And it could either increase stepwise, or it could then approach a certain threshold when it is not healthy and then the person has diabetes, or then the person is treated with insulin, it goes back to the normal levels. This is the, housekeeping conventional understanding of biomarker. Can such biomarker exist for aging, which is a non-linear process? It's not a linear process, right? You don't get a gray head of hair because you get one gray hair per day. One gray hair, 2, 3, and so forth. No, it is clearly, everybody knows it's not a linear process. Furthermore, it is kind of a stochastic dysregulation of various systems. So if you are looking for a signal, but our noise, static, increases with time, we cannot even see the signal because static becomes so loud. So those type of questions, I think, will redefine not only the idea of biomarkers of aging, but probably will prevent waste of time and money on still trying to find them. Going full circle, we didn't find them. What are biomarkers of aging? We didn't find them. What are the biomarkers of aging? And so forth. So So I will give a talk today, and that's what Generation Lab, in fact, one of the focus is on redefining what are the biomarkers of aging from the stepwise change upward or downward to measuring dysregulation or biological entropy or noise or static itself. So I would like to also, I'm passionate about it, to end up on this kind of big question. In order to find anything, including biomarkers, first you need to answer, why do you age? It is true for every disease, right? We found biomarkers very quickly for every single disease. We are not really incapable of finding biomarkers. We found them for every single disease, but not for aging. So the question is, why do we age? And so answering this question, in my opinion, correctly, means that we age because of the second law of thermodynamics, because we live in the entropic universe. But that tells you that you are deteriorating stochastically. And for that, there could not be a biomarker.
Provocative view. What about you, Denis? How do you think about biomarkers in clinical development for aging?
Well, I think that 2 points I would like to make. First of all, I will respectfully disagree that we can find biomarkers for every disease, every single disease. In fact, for some diseases, people have been looking for biomarkers, and osteoarthritis is one of them, and there's still yet to find one. So there's no a single clinical biomarker approved, and we would love to have one. So I think the question is, shall we really look for something that we call biomarker of aging, right? Or we talk about biomarkers of specific age-related diseases, right? That will be a lot more easy for the FDA to understand, right? And it's also the second thing, and this is probably the main thing, is that every biomarker has its probability value, right? It's something that is developed and designed using population health approaches. It's just a correlation thing. It's not personalized medicine. And I think the higher you raise the bar, the more difficult it is to actually create these correlations, right? So if you just need to, as you mentioned, glucose, right? So it's still not 100%. It's a very good biomarker, one of the best. And you know about cancer biomarkers, PCI and all these things. They're not 100% predictive of prostate cancer, for example, but this probability is very high. So more general you go, more difficult it is to establish those correlations, right? So when you go from a single disease and specific biomarker to something like a biomarker of aging, you're just raising your bars to extreme levels. I'm not saying it's not possible, but it's really difficult to hit the statistics and convince the FDA that these correlations that you are finding are sufficient to actually use it for clinics. And the second side of it is the cost, right? So healthcare system will not really deal with biomarkers that cost tens of thousands of dollars per patient to do. And that's what we do in academia. Some of our studies, they're very expensive, but, finding something simple that can be done in a blood test or something is, the cost can be actually also a serious consideration.
Can I?
Please.
Yeah, so totally valid points, right? So I'm not an expert in osteoarthritis, but my husband has osteoarthritis. And it was, now he had hip replacement, so he's much better. So it was super easy to diagnose it. He had pain, they did x-ray, and they found with 100% certainty that he has osteoarthritis. It was not correlation, it was actually causation, because he didn't have cartilage left in his hip, and his bones were grinding to each other, and it's a cause of osteoarthritis. And Denis is completely correct that if you start looking at population statistics and correlations and blood biomarkers, that makes it immensely difficult problem. But we are not talking about that. We are simply talking about what are the biomarkers, not necessarily from droplet of blood. What are the biomarkers of aging? You can look at your retina, you can look at your bone density. There are various approaches. And when we looked, in none of the systems, aging is a linear process. There is no stepwise transition. And in every single system, interpersonal differences are so noisy, particularly at middle age and old ages, that they mask any signal. So that's kind of the second part of the same problem, is that we are not doing correlation. Ideally, we'll do causation. And causation-wise, yes, every single disease can be diagnosed today. And it takes about a couple of years for new diseases to say, this is the gene that causes this disease. And we can diagnose you. It is a biomarker of disease. We don't have it for aging. Personally, my beliefs are for philosophical reasons, that aging is the stochastic deterioration, for which there could not be a stepwise biomarker.
Denis?
Yeah, I agree with that. But I just want to add that I think the main value of having a biomarker, real biomarker, is actually prediction. When you mentioned that when we do X-ray and we see a Colgan-Lawrence stage 4 osteoarthritis, you mentioned at the beginning, right, it's too late. So pain, when our patients have pain, it's too late. And we would love to find something that will tell us that this person actually has a very high risk of developing a severe health decline and disease, and we are looking for that, but to date, we don't have it.
Yes, we do, with Generation Lab. So that's how I would like to end this part of the argument. Exactly the problem that Denis outlined. Before there is pain, before there is clinical diagnosis, what can we do? Generation Lab, first of all, the Conboy lab in the past, and now the science and translation moved to Generation Lab, are focused exactly on that. How can we tell a person before they have pain or symptoms that this body system is being dysregulated to the point that it turns into pathology?
I want to bring this back full circle. I want to combine the predictive question with something we were talking about, which was clinical translation and having FDA allow biomarkers into clinical trials. I'm going to make an example, which is we know of serum markers in the blood that are pretty strongly predictive that someone's going to develop a neurodegenerative condition in the future. For example, Alzheimer's. And they're not 100% predictive, but they're statistically pretty good. And what I have a strong prior belief, and I think it's not uncontroversial to say that it'd be great if we could treat Alzheimer's before it was symptomatic. By the time it's symptomatic, it's probably a little bit too late. Wouldn't it be lovely if we could find interventions that influence these biomarkers that were causative biomarkers that had to do with the cause of the disease early enough that the patient was asymptomatic and remained asymptomatic, right? So that'd be great, right? But right now, that's not how FDA allows clinical trials to design their endpoints. Do you think, and it's, we had a little sidebar earlier, and it's okay if you take your controversial position, Denis, do you think there's any hope that FDA will allow that sort of thing in the next 10 years?
No, there's no hope. But I think what we all should do is to add those biomarkers as part of our clinical trials as research outcomes to convince them eventually that it has value. They're not going to accept it until you show them very strong results and evidence. And unfortunately, at the moment, I only see it being done using retrospective studies, not prospective. So we need to be patient and strategic about it, and I think that's how it should be approached, as including all these sophisticated strategies and diagnostic tools as a research outcome, potentially measures for clinical trials that will still be very conventionally run, and they will not allow to make any claims based on this research outcomes, but I think with growing evidence, eventually there will be a critical mass to change their mind. Not probably in the next 10 years, though.
Okay, so there's hope. It's just not hope for tomorrow or next
year.
Yeah,
I mean, you asked about 10 years.
I did.
Yes, so in 10 years, difficult, but eventually I think there is hope.
Okay, not impossible. And Irina, I'd like to hear Generation Lab's perspective on this
idea.
Yeah,
I'm more hopeful. I think that eventually there will be new longevity drugs on the market that will put FDA on notice that the traditional way did not work very well, and what was approved by FDA to be tested is lackluster. whereas other approaches are working more effectively. And so I hope that will be an inflection point, and also with the help of these groups. For example, Dylan Livingston at the helm of A4LI, and Brenda and all of us. I personally think that we should just be more vocal about it, and we should highlight what is already succeeding, because it would be very difficult then to fight against success and say, no, let's use our traditional ways, even though many of the drugs which now companies spend hundreds of millions of dollars and are on the market are not performing well enough.
Yeah, I think one aspect may help us. It's the fact that we deal with aging population and the healthcare systems collapsing everywhere. So if you look at the economics of Medicare or some other things that we are dealing with now, and in our field we have one total joint replacements a year now in the country, and they're projecting it to be 1 million, and now jumping to 3 or 4. If you talk to people who are dealing with hemodialysis, I mean, Medicare is pretty much done. It's not ready to support it even now. Alzheimer is another thing. So they may be forced economically to go into preventive strategies because they just cannot afford dealing with this late-stage diseases. And it may actually help us in a very unusual way, is that they will just need to deal with economics and with the budget deficits that way.
I completely agree. And I think FDA actually did a marvelous job on the whole field of biomedicine in getting rid of so many diseases that used to kill people before they were 40 years old. But ironically, that now brought up all of these age-related diseases that we cannot cure. And that, of course, is a burden, like really unsolvable burden. And so that, again, as Denis mentioned, I'm just summarizing his point, might move FDA towards the realization that the former approach now is not working anymore and change the strategy. And I think we should all just advocate for that. Say, okay, guys, you did a great job. Now we have age-related diseases. Population of the planet is getting older. Diseases are chronic. And there are no systems in place. There are no socio-economical systems in place to make it sustainable. What do we do next?
I think those are both fantastic closing statements. I was going to try to evoke one, but you did it unprompted. So everyone, please thank our panelists. And I'll hand the microphone back to Brenda. Oh. Well, my watch is a little fast.
Yeah.
All right, yeah.
We can have a mic.
I can also yell. I don't mind. I'm loud. So, hey, thank you to everyone on the panel. That was a great discussion. So, Ben from Oro Biomedical. So we spent a lot of time talking about the FDA because that's where we're developing our drugs in this ecosystem. Do you see any other countries' regulatory bodies being more amenable to aging and treating aging and looking at biomarkers of aging? Is there any way to evoke pressure by saying other people are doing it?
Well, I think it depends what your goals are. If you just want to make money, yes, this may be a good strategy. If you want to convince the FDA, I don't think it will help much if you go to Qatar and have your drugs approved there. So it depends on, again, what you want to achieve with these foreign regulatory agencies.
I'll jump in and say it depends on what you mean by country. There are certainly, we all know, I'm not going to mention them by name, but certain kind of extra-national initiatives trying to create special zones. I'm not going to take a position on that right now until you put a couple drinks in me. But I will say that I think that sometimes those amount to a kind of epistemological end run around what we're actually trying to do that benefits patients in, I'm taking a position, aren't I? And so, you end up not doing certain tests when you liberalize the regulatory regime. So anyway, I guess you guys know how I feel about this now.
Thanks so much. This is Irina Bargava. Great discussion there, Irina and Denis. I think that's a really relevant point, that FDA has a responsibility, and then we as consumers, scientists, and researchers also have a responsibility to build things together in a scientific fashion. So point being, when we're talking about randomized controlled trials and we choose an outcome, we do have to choose the primary outcome, and then we decide a sample size for that. We can have a huge number of secondary outcomes, but now we need a sample size for that, which obviously increases the cost, increases the duration of the study, and the feasibility overall. And therein lies the huge problem. So now if we do a meta-analysis and put all of them together, it's apples to pears. Everybody looks skeptically at the results of a meta-analysis, but there is value there. So I think as folks are building tests and therapeutics, our responsibility is to try and see as much as possible that we include the additional outcomes markers that we think bring all of us to a common platform. So there isn't one new methylation test one day and 3 weeks later somebody else says mine is better. At the end of the day, we've got to bring it to the populations. We've got to bring it to the clinics. It cannot just stay in the point-some% population that's ready to be biohackers, concierge medicine. And for that, we do need this, responsible effort, and FDA is going to back it. Once they get the evidence, they will back it.
And this gets right back to what Denis was talking about accumulating evidence over time.
Yeah, and I think you are absolutely right. It's a fantastic comment about the statistical aspect of it. It probably can be only done during phase 3 trials. It's very difficult to hit anything meaningful before that. But what we know, and this example that I gave about cardiovascular trials and then unexpectedly hitting positive results for osteoarthritis, those were phase 3 trials, and you need probably thousands of patients to catch that.
I would like to actually interject and present an opposite point of view. I think that medicine that truly works does not need that, does not need thousands of people and various parameters and multivariable statistics. If it does work, if a person loses weight and becomes younger or healthier in some ways, like GLP-1, it works. And it is robustly obvious from simple clinical trial. If it works a little bit for some population and perhaps does not for the primary parameter but works for the secondary parameter, completely true. But if you are looking at the breakthroughs in aging and longevity, that you are getting older and now you stop getting older, that will be abundantly clear on multiple redundant assays that could be done with relatively few people and will not invoke statistics. It will really be direct measurement with normal things like mean, standard deviation, and p-value.
So what I hear you saying, Irina, is just have such a big effect that you don't need to do statistics
Then
on
you
your
don't
study.
you always need to do statistics, but you don't need complex statistics and very expensive trials to come to a conclusion or convince anybody. So Tom Rando, who is not here today, right, he taught me back when I was being postdoc that there are 3 R's that are important in biomedicine, that it should be relevant, robust, and reproducible. And that's it. So if it is relevant for a disease or several diseases, robust, so you really change situation from being less healthy to much healthier, like GLP-1 or insulin, and reproducible, so it always works. Then statistics become simpler, and then the trial shows success much faster.
To your point, that's the effect size that we factor
Effect size, exactly, it's effect size.
into calculating what the sample size we need.
Sure, exactly.
Size is huge. You need a smaller sample, but again, you're doing rigorous research to prove the point.
Yeah, so another metaphor, right? Let's say that you are given 2 different glasses of lemonade and you need to figure out from which stand. Difficult to do, you need to drink many glasses and you need to analyze taste receptors. If you are given a glass of lemonade and a glass of seawater, you only will make 1 sip. You say, I'm not drinking anymore. So that, yes, officially it's called effect size and all of that. But in reality, for any meaningful longevity therapeutic, we do not need complex clinical trials.
As an employee of a drug development company, may I say from your mouth to God's ears that we all have such great effect sizes that we don't need to have large sample sizes. Do we have time for one more question, Brenda?
Yeah, we have another one here too, but go ahead.
Okay.
Hi, myself Dr. Jamal. I'm a former professor in cancer research, and I'm building Vitalistic, all-in-one AI-powered longevity and wellness platform. And I just want to be, like hopeful in a way of you commented about it will take 10 years for FDA to convince, and maybe more than that. I believe in the age of AI, we should be pretty more hopeful in a way it should be sooner. And we should make our environment being a responsible, in this segment, how to educate them. Sometimes, I believe FDA is still working in the age of old age kind of a style, and possibly they need to bring some kind of reformation where they can expedite things in a way which is helpful to the people at the mass level, not just for the niche people in a particular segment of longevity and wellness is people think this is just for few segment of rich class people are having it. I believe this is our responsibility is to bring it to the mass level, how everybody can achieve that, in their lifestyle. Thank you very much.
Yeah, I'll just give you one example. In our field, we are trying to convince the FDA to accept MRI as a valid outcome measure for joint osteoarthritis, with thousands of publications being done showing sequences being coupled. So it's taking 20 years and we are not still there. It's a simple imaging technique for knee joints for a very clear disease, and it's taking decades to convince them. So what you are talking about is a completely different magnitude, and the challenge is much higher. I'm with you. I hope it will be accelerated, but we need to be realistic here.
Yes. Hi. So I liked what you had to say about cats and dogs potentially being a way forward for exploring some of these ideas. I come from a pet industry background, so I'm curious, what are you seeing in the cat and dog and veterinary space that's most exciting to you right now? And and is it really applicable to humans? Because they are, even though we think of them as little people, they're still different species.
Yeah, I think that there are different species. And also mice age very similarly to people as well. So I'm not 100% sold on the idea that if we stop doing research in mice and start doing research in dogs, or cats, we will discover something different. However, we might discover some details that mice do not provide, and that will allow us to treat human diseases better. On the other end of the spectrum, since I'm from College of Engineering, Department of Bioengineering, there are also human organ chips, which are not cats or dogs or mice. And they are not just cells in culture, they are much more sophisticated microphysiological systems, which I think will be another option to expedite success of clinical trials.
Yeah, I think this is a great area. There are plus and minuses. I think the major plus is that there are hundreds of millions of companion animals, and you can get a great statistics and involve a lot of these dog owners in the trials. They all want their pets to live longer. And regulations are not as strict, obviously, as for humans. You can enroll, you can claim health span extension in animal space. There's no problem. You can actually market drugs and loyal this company from Bay Area, that's what they're doing. So if you do it for dogs, and we do hundreds of thousands of pets and show it to the FDA, because they are evidence, they're reasonable people, they need arguments, right? And you show that you extend health span, it will be very appealing to them. And I think that's what we're all doing, because on average dogs, they live much shorter life than humans. right? On average, they live 10 years, and in 1 year, you can capture things that was a huge problem in humans, that all these biomarkers require 20, 30 years to develop, right? It can be a lot faster in dogs. The downside is that they don't develop diseases, exactly the same way as humans. For example, dogs never have ischemic heart disease or stroke, right? Because they're carnivores, they eat meat, but they don't have atherosclerosis. They only die of cancer and some other things, but humans, 50% of humans will be killed by ischemic heart disease or stroke, right? So some of these diseases cannot be modeled. So there's some limitations, but I think there's some value there as well.
All right, that was a great panel discussion. Thank you, Chris, Irina, and Denis.